Imagine a microscopic instruction manual coiled up inside almost every cell of your body, containing the essential information that makes you, you. This isn't science fiction, it's DNA, the astonishing molecule that holds the blueprint of life, guiding the development and functioning of every living organism. DNA stands for deoxyribonucleic acid, and it's responsible for the incredible diversity of life on Earth, from the simplest forms to the most complex. From the smallest bacteria to the largest whale, DNA orchestrates the development, growth, and reproduction of all living organisms, ensuring that life continues to thrive and evolve. Think of DNA as a recipe book, passed down through generations, containing the secrets of life. It contains all the instructions needed to build and maintain an organism, ensuring that each new generation inherits the traits of its predecessors. These instructions are organized into genes, which are like individual recipes for specific traits like eye color or height, determining the unique characteristics of each organism. Understanding DNA is like unlocking the secrets of life itself, revealing the intricate details of how living beings are formed and function. DNA's story is one of remarkable complexity and elegance, a testament to the wonders of nature. It's a tale of how simple chemical building blocks can encode vast amounts of information, shaping the tapestry of life as we know it, and continuing to inspire scientific discovery and wonder. Zooming in on DNA, we encounter its iconic double helix structure, resembling a twisted ladder. This intricate design is not just visually stunning but also fundamental to the molecule's function. This elegant architecture is formed by two long strands that spiral around each other, creating a stable, yet flexible framework. Each strand is made up of repeating units called nucleotides, which are the essential building blocks of DNA. Nucleotides are the building blocks of DNA, and they play a crucial role in the molecule's structure and function. They consist of a sugar molecule, a phosphate group, and a nitrogenous base. These components are meticulously arranged to form the nucleotide. The sugar and phosphate groups form the backbone of the DNA strand, providing structural integrity, while the nitrogenous bases stick out from the sides like rungs on a ladder, ready to pair with their counterparts. There are 4 different nitrogenous bases in DNA: adenine, A, thymine, T, guanine, G, and cytosine, C. Each base has a unique shape and chemical properties. These bases pair up in a very specific way. A always pairs with T, and G always pairs with C. This specific pairing is known as complementary base pairing. This precise pairing is crucial for DNA's ability to store and transmit genetic information. It ensures that genetic instructions are accurately copied and passed on during cell division, maintaining the continuity of life. The sequence of these base pairs, AT and GC, is what makes up the genetic code. This genetic code is the blueprint for all living organisms, dictating everything from the color of a flower to the structure of a human brain. This code is like a language, with each 3-letter word known as a codon, representing an instruction for building a specific protein. These proteins are essential for the myriad of functions that sustain life. Proteins are the workhorses of the cell, carrying out a wide range of functions, from building tissues to fighting off infections. They act as enzymes, hormones, and structural components, playing a crucial role in maintaining the health and functionality of an organism. Just like the 26 letters of the alphabet can be arranged to form countless words and sentences, the 4 bases of DNA, adenine, thymine, guanine, and cytosine, can be arranged in countless ways to create the diversity of life we see around us. This incredible variety is what allows for the vast differences between species, as well as the unique traits within a species. The human genome, for example, contains over 3 billion base pairs, which code for an estimated 20,000 to 25,000 genes. These genes are like individual instructions in a massive manual, each one contributing to the complex machinery of the human body. These genes determine our physical characteristics such as eye color and height, as well as our susceptibility to certain diseases. They influence everything from our metabolism to our immune system, playing a key role in our overall health and well-being. They are passed down from parents to offspring, ensuring the continuity of life. This genetic inheritance is what connects us to our ancestors and will link us to future generations, carrying forward the legacy of life. For life to continue, cells must divide and create new cells. Each new cell needs a complete copy of the DNA instructions. This is where DNA replication comes in. Imagine the DNA ladder unzipping down the middle, breaking the bonds between the base pairs. Each separated strand then serves as a template for building a new complementary strand. Free-floating nucleotides within the cell match up with their corresponding bases on the template strands, A with T and G with C. This process is carried out by a team of enzymes which are like tiny molecular machines. The result is two identical DNA molecules, each consisting of one original strand and one newly synthesized strand. DNA replication ensures that each new cell inherits a complete set of genetic instructions. Section 5, From DNA to RNA, Transcription Explained. While DNA holds the master copy of genetic instructions, it's not directly involved in protein synthesis. Instead, DNA's information is first transcribed into a related molecule called RNA, or ribonucleic acid. Transcription is similar to DNA replication, but instead of creating a new DNA molecule, it creates a single-stranded RNA molecule. The DNA molecule unwinds and one strand serves as a template for RNA synthesis. RNA nucleotides pair up with their DNA counterparts with one important difference. Instead of thymine, RNA uses uracil. So the base pairing rules for RNA are A with U and G with C. Section 6, RNA's role carrying the genetic message. Once the RNA molecule is synthesized, it detaches from the DNA template and carries the genetic message from the nucleus, where DNA resides, to the cytoplasm, where protein synthesis takes place. This messenger, RNA or mRNA, carries the genetic code in the form of codons, which are three base sequences that specify which amino acid should be added to the growing protein chain. Think of mRNA as a messenger carrying a coded message from the DNA blueprint to the protein-building machinery of the cell. Section 7, Protein Synthesis, Building Blocks of Life. In the cytoplasm, mRNA encounters ribosomes, which are the protein synthesis factories of the cell. Ribosomes read the mRNA codons and use this information to assemble amino acids into proteins. Each codon corresponds to a specific amino acid. For example, the codon AUG codes for the amino acid methionine, which is always the first amino acid in a protein chain. The ribosome moves along the mRNA molecule, codon by codon, adding the corresponding amino acid to the growing protein chain. This process continues until the ribosome reaches a stop codon, which signals the end of the protein. The newly synthesized protein then folds into a specific three-dimensional shape determined by its amino acid sequence and carries out its designated function in the cell. Section 8. The Diversity of Life, a Symphony of Genes. The remarkable thing about DNA is that despite its simple structure, it can encode an incredible diversity of life. From the tiniest bacteria to the largest animals, all living organisms share the same basic genetic code. The diversity of life arises from the different ways that genes are expressed and regulated. Genes can be turned on or off, and their activity can be increased or decreased in response to environmental cues or developmental signals. This intricate interplay of genes and environment creates the incredible tapestry of life we see around us. Section 9, Genetic Variation, Why We're All Different. Even within a species, no two individuals are exactly alike. This is due to genetic variation which arises from mutations or changes in the DNA sequence. Mutations can be caused by errors during DNA replication, exposure to radiation or certain chemicals, or even just random chance. Most mutations are neutral, having no effect on the organism. However, some mutations can be beneficial, providing an advantage in a particular environment. These beneficial mutations are the raw material of evolution, allowing populations to adapt to changing conditions over time. Section 10. DNA Technology, Unlocking the Secrets of Life. Our understanding of DNA has revolutionized biology and medicine. DNA technology allows us to study genes, diagnose diseases, develop new therapies, and even modify organisms for specific purposes. DNA sequencing, for example, allows us to determine the precise order of nucleotides in a DNA molecule. This information can be used to identify genes, diagnose genetic disorders, and trace evolutionary relationships. Gene therapy holds the promise of treating genetic diseases by replacing faulty genes with healthy ones. And genetic engineering allows us to modify the genomes of organisms, for example, to create crops that are resistant to pests or diseases. Section 11 Conclusion DNA, the blueprint of us. DNA is the blueprint of life, containing the instructions for building and maintaining all living organisms. Its elegant double helix structure, precise base pairing, and intricate mechanisms of replication, transcription, and translation are a testament to the power and beauty of nature. Understanding DNA has revolutionized our understanding of life, from the molecular level to the grand sweep of evolution. As we delve deeper into the mysteries of the genome, we continue to uncover new insights into the complexity and wonder of the natural world. From the smallest bacteria to the largest whale, DNA connects all living things, reminding us of the interconnectedness of life on Earth.